We are driven by an interest, which is to understand the evolution of the brain. And of course, that's a big, big question to ask. And so I would rather reframe it as saying, we want to understand general principles or get some ideas of how nervous systems and brains in particular achieved their incredible complexity, which enables us to do all the wonderful things we do in terms of like math, music, and language reasoning, and all the products of our intellect and imagination. But of course, the brain, the story of the brain didn't start with any of that. As Lynn mentioned, I did my PhD studying the brains of small marine worms and appreciating the simplicity of nervous systems that live, in that case, in the ocean. And understanding the evolution of the brain is about piecing together that journey that goes from simple to many different and independently evolved iteration of complexity. Before diving deeper into this matter, I want to use a metaphor to illustrate why the brain does not evolve simply following principles of optimization and optimal design, but why exactly the historical approach is in comparative approach that I'm going to talk about is needed to understand some of the peculiarities of the structural and functional organization of the brain. So this is an image of a square, very famous square in Rome, Italy. I come from Italy, as you've heard. And the first time I went in there, I thought, this place makes no sense. You know, a square is a place where you go to meet other people, to gather, and to spend time together. But if you enter a square like this, which is so elongated, and please compare with the cars on the side, to appreciate how long this is, there's no way, if you enter on one side, to see who is on the other side. So this place does not serve the function of a square. Nobody would come up with such a design if they wanted to build a place for people to meet and encounter each other. But the reason why this place has this shape is because it used to be a stadium for horse races built by ancient Romans. And so what happened is that this stadium, obviously if you want to have horse races, you need to make very long. This stadium was reincorporated in the fabric of the city over the centuries, and that's what gave this very peculiar place to Rome to these days. So I think this is a metaphor of what happens and what happened with the evolution of the nervous system. In many cases, and that's what we are finding over and over as we dig into the details, elements are repurposed for new functions. So elements being cell types or neural circuit motifs, sometimes even entire brain regions, or gene regulatory networks. And it's important to appreciate how frequently this happens because it is a manifestation of the unique contingencies and constraints that operate on the evolution of the nervous system. But also understanding these processes helps us understand some of the odd features that the brains can have in terms of their structural functional properties, which are, again, not the result of principles designed or optimality. This is not how our brains came to be. And when I think about the evolution of the nervous system and of brains, what strikes me as the bigger question that we still have not been able to answer is how did innovations arise to confer animals, like the ones you see on the screen, their incredible cognitive abilities. You're seeing, primates with their tool use, and same as octopus and cephalopods that are very complex lives and minds, and birds. And these are all instances of independently evolved behavioral sophistication. You may want to call it intelligence. We can discuss the definition of that. But we do not have, as evolutionary comparative neuroscientists, a good principle explanation of what makes these brains truly different from the brains of their closely related species. Do you need to have more cells to be smarter? Do you need to have more connections? Are there, for example, certain gene families that expand to make brains more complex and behaviors more sophisticated? To tell you the truth, I don't think we have an answer to any of that. What we're missing is principles, ideas, and mechanisms of how novelties arise in the brain and the nervous system. And just to build this contrast, I want to compare with what is known from other fields of biology. For example, in genetics and genomics, we know that one of the many mechanisms to evolve new genes is by duplication and divergence. You have a gene, that gene duplicates. At that point, that there's a release of selective pressures on one of the copies, and then that can freely evolve to acquire a new sequence and function. In evolutionary developmental biology, EVO-DEVO, we've learned in the last 20 years the new body parts evolved through changes of regulatory programs that are active during development. This is just one of the many examples where you see how regulation of certain genes correlates with changes in the morphology of appendages from fins to limbs in vertebrates. There's countless examples of that, so I would call that a principle for explaining the emergence of novelty of body parts during evolution. We do not have principles like this for the brain, and that's what we are striving to learn by comparing nervous systems in my lab across vertebrates, and particularly tetrapods, and trying to find similarities in differences in instances of innovation. What makes it so hard for the brain is that the brain has a multi-scale organization. So you can think of many instances of places where these innovations may take place, right? You can imagine that new genes evolve and confer new properties to certain neurons. You can imagine that even the genes that you already have there start interacting in different ways, and those new interactions give new properties to neurons or entire parts of the brain. You can imagine that these new interactions, for example, lead to the emergence of new types of neurons that were not there before, and these new types of neurons again play some important function confer new functions to these brains. You can imagine that neurons change their connectivity. Even neurons that are already there, they start to connect and interact in new ways, and that again might be a source of innovation. And then you can also speculate. There's entirely new brain regions that are involved in these behaviors. And so things at the very high level we want to understand is, are certain types of novelties more likely than others? Are certain brain areas or circuits or neuron types more evolvable than others? Is every type of cell equally likely to change over time, or are there constraints that selectively limit the evolution of the brain and force the evolutionary path to take certain directions rather than others? And finally, and the bigger question is, how do these different structural innovations that we can imagine that play out, how do they impact function? Are these expanded behavioral capacities of certain animals the result of certain specific types of structural innovations, or can it strike anywhere? And so these are the broad questions, and more into the specifics. We're looking at this part of the brain, the neocortex, because, as the name says, it's been defined as a new region of the mammalian brain. It takes more than 70% of the volume of our own brain. Is essentially the most superficial part of the rough part of our brain. And it is considered an innovation because of its architecture. It's organized into 6 layers of neurons, which you see in this schematic here. And these 6 layers have been classified on the basis of the position and morphology and the properties of these neurons. Zooming in, these are some reconstructions of the kind of neurons that you find in a mammalian, in this case a mouse, neocortex. These neurons have distinct molecular identities, so they express different genes. They have different morphologies. They have different connectivities. And because they connect with one another and with neurons outside of the neocortex in different ways, they also play different functions. So in other words, understanding the origin of all these different neurons in the 6-layer neocortex is critical to understand the functions that this part of the brain is involved in, which is processing of very high level information, sensory information, integrating sensory information, involvement in learning and memory, and many other things. So how do we go about that? Being the neocortex a mammalian innovation, we want to compare the mammalian brain to the brains of other vertebrates. And that's what my lab has been doing in the last few years. And actually in this presentation, I want to bring together a synthesis of what we learned from the moment I started working on these questions as a postdoc, which was 10 years ago. And so I will tell you about amphibians, the salamanders that you see on the screen, which are the main system that we have in the lab at the moment. But I will integrate all of that with observations that come from previous work on reptiles, on turtles and lizards. And why looking at these animals? Because they are the closest relatives to mammals, but also because altogether these animals are tetrapods. So these are the animals that adapted to life on land. And it is believed that the transition from water to land was a pivotal moment in vertebrate history that triggered a whole series of innovations in body physiology and the sensory world that these animals were exposed to, and so we think also in the brain. I will focus on this part of the brain, which is the telencephalon. You see here in blue is the most anterior part of the brain. The cortex and the neocortex in mammals belong to that. And I just want to give you a sense of the incredible diversity in morphology in the telencephalon. I will now show in the next slide a section through the telencephaly of some of these animals. And I hope you can appreciate how different these are. These are not in scale. If they were to be in scale, you will not even see the salamander. And you can see a wide variety of morphologies, from layered structure to structures that look more like nuclei. It's been very hard, and this is the history of comparative neuroscience, to make sense of this diversity and to compare things with one another. And zooming into the cortex, I'm going to compare the 6-layered neocortex of mammals with cortical regions or pallial region. This is a term that we use to refer to the dorsal telencephalon in reptiles and in amphibians in the most dorsal part of the telencephalon. And as you can see, these are the definitions that have been given by classical work on comparative neuroanatomy. The reptilian cortex is considered 3-layered because it's time to use the stick. Because it has one layer of neurons, you see the cell bodies here in black, sandwiched between 2 layers of processes. And instead, in amphibians, you don't have that layered organization. So apparently for neuroanatomists, that was enough to say that this is not a cortex at all. And so, of course, when you look at this diversity, you ask yourself how to compare these brains to one another. And many approaches have been used. But what has been quite informative for us and for the field, I would say, the last 10 years, has been to look at neuron types as units of comparison. Why neuron types? Because you can see them, as this schematic is trying to illustrate, as the building block of the brain. But also because we can very nicely define neuronal identity as the combination of the expression of transcription factors that control gene expression in neurons, and the genes that are downstream those transcription factors, which we call effector genes, and are the genes that are involved in playing functional roles in the cell, like they are the ion channels, the neurotransmitter receptors, the axonal guidance cues, and all the things that make a neuron and make neurons different from one another. And so given that definition of neuron type, which is purely molecular, but that applies to other definitions that you may think of, because all these effector genes correlate with different aspects of neural identity, like function and morphology and neurotransmitter. Given that definition, then we can also very nicely scale up the characterization of neuron types by looking at gene expression in individual neurons, in individual cells. And this has been made possible exactly 10 years ago, I believe, by the invention of a technique called drop-seq. It's a technique that it was one of many iterations, but it's a technique that enables us now to capture neurons or cells, actually, one by one, and then tag these cells and then sequence all the messenger RNAs, which we call the transcriptome, of each individual cell one by one. And this is how the data look like. In this matrix, you see every pixel is a gene expression value in one cell. So columns are neurons, rows are cells, and gold means that the gene is expressed in that cell and in that neuron. So these are massive data sets. We're talking about the 20,000, more or less, genes in a vertebrate genome, and thousands and thousands of neurons that we can sequence one by one. And then what we do when we collect this data, we do some analysis, and what we want to find out is what are the neurons that look like, the single neurons that look alike, and we call them clusters because we do clustering of gene expression data, and we use these clusters as a proxy for cell type. And another way to look at these clusters are plots like this. These are dimensionality reductions where every dot is a transcriptome, is a single cell, and the proximity of dots indicates similarity of transcriptomes. And then we look, we cluster them, and we represent the clusters with different colors. And this is the first data we collected on turtles many years ago, and then we expanded to lizards and to the salamanders. And a lot of our work has been about comparing these data sets to data that have been released by other people in the community, especially in mouse. And here comes the hard part, comparing. You see here different colors on the screen. That's because it's not trivial. They're not matching with one another. It's not trivial to say what cells correspond to what. And so we've used, and you'll see examples today, different methods to compare these transcriptomes. We can look at marker genes, our favorite genes, and see whether they match up. We can correlate gene expression across clusters and say, okay, this at the whole clustering level, at the whole transcriptome level, these look alike. Or what you see at the end, we can do some more sophisticated cluster-independent agnostic analysis where we bring all these cells together in the same high-dimensional space. We analyze them all together, all the cells from these different species. And then we see what cells from what species cluster together with one another. And of course, when we do these experiments with single-cell RNA sequencing, we take the brain and we mash it into soup. So we lose all the information about where these cells are in the brain exactly. And so to reconstruct that information, what we do is to look for marker genes that are specific for each cluster and cell type and stain brains with those marker genes. And you see an example of a salamander, 4 brains stained over there. And actually, with the salamanders, this has been very nice because we can do that at scale. Their brains are so small that we can stain the entire brain and then make it transparent and use light sheet imaging to get the entire brain volume and have a complete overview of where these genes are expressed in these brains. And this part of our pipeline is what takes the most time to make sense of these data, but it's crucial because it's part of what we want to find out is not only how neurons compare to one another, but also where they are and what that really means. So for the rest of the presentation, I thought I would go through some of our data to put together, to distill out some of the lessons and the principles that came out from all this work. And I will go back to this and break down the comparison of neuron types across these tissues into comparison of the 2 main classes of neurons that you will find in a cortex, excitatory and inhibitory neurons. The excitatory neurons are glutamatergic, are the ones that define the layers, are the ones that are different across the different layers. And the inhibitory interneurons are GABAergic, and they're all scattered around. And before we were us being able to analyze and compare these neurons at the single neuron level, there were many ideas on how these brains will be related to one another. And the most popular ideas were about the turtle layer being homologous, so corresponding to either the deep layers or to the superficial layers of the mammalian neocortex. So the idea was that there would be some clear, one-to-one correspondence of the turtle neurons or the reptilian neurons more in general to certain layers of mammals, and then other layers of mammals would be new and innovation. And then the other idea was that the same would be true for interneurons, that certain interneuron types would be mammalian specific and other interneuron types would be instead more ancient. And we found that none of this is really true. So for the GABAergic interneurons, what we saw when we just looked at the simple list of mammalian marker genes, these are the marker genes that are sufficient to classify mouse neurons. You're seeing here the genes in rows, the neurons in columns. These are genes that are sufficient to classify mouse neurons into 3 general classes. And when we take the same genes and we ask where are they expressed, how are they expressed in a turtle, in a reptile, we find that these genes are sufficient to define the same 3 classes of interneurons. And so that was very surprising, and that was already an indication that these interneuron classes may trace back to more than 300 million years ago in evolution. And so then when we did the same analysis for the other class, the glutamatergic excitatory neurons, and so we took all the genes that define the different layers in mammals, and we looked at them in the reptile, over there. And we saw that here the picture is very different, right? So you have genes that are expressed in very specific combinations in the mammalian layers that are instead expressed either everywhere or nowhere in the reptilian cortex, indicating that these reptilian glutamatergic neurons are something completely different. And so to get a better understanding of what they are, a few years later, we collected data also from the salamanders, and we started using these data integration techniques to have a more unbiased and systematic analysis, going away from the selection of specific markers. And here I'm showing you all the data from these different species all together in this plot. This is probably the most complex data slide that I have. And the bubbles here are clusters. And in a moment, I'm going to color these clusters by species mixing. As I told you, here we analyze all this data from different species together with some methods and considerations on how to do that properly. And now I want to know if for each of these clusters, what's the relative contribution of cells from the different species that I used for the analysis. So you will see, in a moment, you will see gray in the clusters that are made of cells from all these different species. And let's say cyan for the clusters that are made mostly of mouse cells, and so on and so forth. And so you see like many colors. Breaking this down, we find that in the interneurons, most clusters are made of cells that come from all these different species in the analysis, supporting the idea that, again, these interneuron types and classes are very ancient, with a few exceptions. But for the excitatory neurons, the glutamatergic neurons, we see that the neurons that belong to the neocortex in this data set, they mostly cluster on their own, as you can see here from the cyan color, indicating that the neurons of the 6 layers of the mammalian neocortex have their own unique molecular profiles that have nothing to do with neurons that we have analyzed and sequenced from reptiles or from amphibians. And summarized here. And so what these reptile and amphibian neurons are, they actually tend to cluster together with mammalian cells sampled from the piriform cortex, which is an olfactory cortex, the hippocampus, the subiculum, the entorhinal cortex, so cortical areas that are not the neocortex. And I should maybe pause here for a second. Some of these cortical areas are also 6-layered. So, for example, the entorhinal cortex is. So the 6 layers are not really exclusive of what we call neocortex. But there is something unique about the neocortex, which is the way sensory inputs are integrated and processed, which is very different from the kind of processing that happens in the entorhinal cortex, which is part of it is closer to the olfactory system and part of it to the hippocampus and involved in navigation and learning and memory. So I think this starts to give us a hint of what, or hypothesis at least, testable hypothesis of what these normal mammalian brains may or may not do with their little and simple cortices. But so the first take-home message from all this is that unlike what was expected from previous ideas and hypotheses in the field, actually the neocortex is not entirely new, but there are some elements to it, specifically the GABAergic interneurons, which are very ancient and trace back to at least the tetrapod ancestors. And so that indicates that brain regions are not just evolving as a whole, but it's important to look at that granularity at the cellular level to understand what's new and what's old. And of course, there's now another question that comes up. Okay, if these neocortical neurons are new, how did they come about? What were the changes that really led to the expansion of neuronal diversity along the mammalian line of evolution and to the invention of new neurons? And our entry point into this was a quite surprising observation that came, again, from the careful mapping of clusters and cell types on anatomy. And I'll start with the turtle again, where we saw that in that cortical region we've been studying, there were neurons that belong to two clusters. And these two clusters are very different, as indicated from their different position in the UMAP space. And they map to two different layers of neurons. So essentially that single layer of neurons can be broken down into two layers, which are not recognizable just by looking at a nuclear staining, but are clearly recognizable by looking at specific marker genes. And surprisingly, but perhaps not that surprisingly, the same holds true also for the salamander, for the amphibian, where you see, where we found, again, two separate layers of neurons here stained with different genes, but actually there's hundreds of genes that are differentially expressed between these two layers. And so this is important for two reasons. One, it pushes back the origin of layered cortices, at least to the tetrapod ancestors. So a layered cortex is not an invention of mammals, but exists for a very long time. So what happened in mammals is that just more layers were added, but the true layers of neurons, the different types of neurons were already there. And second, it tells us something more general, which is how important are these molecular tools to really understand the complexity of the anatomy of these brains, because there are so many traits that are just hidden and cannot be recognized by looking only at a nuclear stain. This was finding these layers was a starting point for or a strong motivation for a project in my lab. Where we decided to dive deeper into the development of the salamander dorsal telencephalon, dorsal pallium. And the reason I think is captured by this slide. This is unpublished data, actually. And what's commonly believed is that in non-mammalian vertebrates, neurogenesis is very simple. It's believed that there's only one type of neurons, that there are no layers, and I just showed you that that's not true. And that these neurons are made by radioglia cells that just divide and make neurons. And that there's a whole series of things, is believed that there's a whole series of things like multipotent progenitor cells, which we call radioglia, intermediate progenitors, that have evolved in the mammalian line of evolution. But of course, these ideas don't really match what we see now with the molecular analysis of these animals. And so that's why we decided to ask, are these principles of cortical development that have been elucidated in mammals, do they extend to the salamander? And so we asked three questions here. One is, are the superficial and deep layers that we found, these two different layers that we find in the salamander, are they generated sequentially over time? And to answer that, we did something very simple, which was to give EDU, which is a marker of cells that are dividing, so cells in the S phase of the cell cycle, at different time points during development. And what you're seeing here is a chart that illustrates the experiment. And these numbers here correspond to developmental stages that have been defined in the literature by people before us. And they're separated by about 2 months. So development is extremely slow in these animals. And when you go from stage 37 to stage 55, it takes about 2 months. And so we've given EDU at these different stages, and the idea is that EDU would be, at the end of the developmental phase, would be retained, would be visible, only in the cells that have exited the cell cycle or have born at the time of EDU administration. And so this is what we saw. We saw that EDU given early labels superficial neurons, EDU given in intermediate stages labels deeper cells that are a bit deeper, and EDU given very late labels cells deep into the pallium, deep layer neurons. So this shows that there is a link between the cellular neuronal birth date, the identity, the molecular identity of a neuron, and its position in this layered pallium, which is exactly the same thing that you will see in a mammalian neocortex. It also shows that neurogenesis in this system is outside in, as we call it, meaning that the early-born neurons are outside, are on the surface, and the late-born neurons are inside. And this is, and we'll come back to that, this is the opposite of what's found in mammals. The second question that we asked is, are these progenitors multipotent? So is there one progenitor that makes the superficial neurons first and the deep neurons later? Or are there different progenitors that are just dividing at different time points during development? And so this is how these progenitors look like, and we can target them with experiments called electroporations, where we insert genetic material into these cells during development. And when we do an experiment like this, we can then use DNA constructs to integrate in the genome of these progenitors a reporter, like GFP, or a library. And in this case, we did an experiment with a library where GFP is coupled with a barcode. So this library has a great complexity, millions of different sequences, which means that this construct gets integrated into progenitor cells, and each progenitor cell gets a unique molecular label. And what we do then, and this is how these brains look like a few weeks after this electroporation, what we do then is to take all the GFP positive cells that have that molecular barcode, that molecular label, and we isolate them, we do single cell RNA sequencing, and we sequence their transcriptome to know who they are, their identity. But we also sequence the barcode with the hope of finding cells that share the same barcode, because whenever we find cells with the same barcode, what that means is that those cells came from the same progenitor. And this is actually, took a very long time to set up. It's an experiment we were fortunate to do in collaboration with Christian Meyer, who was the one that invented this technique called tracker-seq. And what we found is summarized here. We found a significant fraction of clones of cells that shared barcodes for superficial cells and barcodes for deep layer neurons. So that's the demonstration that the progenitors are multipotent, again, like what you would see in a mammalian cortex, and those multipotent progenitors make neurons over time. And finally, the final question we went on to ask was, do these progenitors exist in different molecular states over time? So the motivation for that question comes again from mouse data, where people have shown in many different ways that progenitors transition from an early state, an early molecular state, to a late molecular state. And these two states correlate with the production of early-born neurons and then late-born neurons in the mouse neocortex, which are neurons in different layers. And so to answer that question, we collected single-cell RNA sequencing from the developing salamander, and we isolated the radial glia progenitors, and what we found was that the same genes, or many of the same genes that define these two states of progenitors in the mouse, also define two states of progenitors in the salamander. So indicating that, again, like in the mammalian system, there is a similar transition of progenitors from one molecular state to the other molecular state, and that transition correlates, at least, that's what we can say now, with the switch from production of early-born neurons, superficial neurons, to production of late-born neurons. And so to summarize this part, we have basically, by diving deeper into the development of the salamander system, we've basically rewritten the story of how these cortices develop. And instead of being something simple and boring, it's actually a system that has many similarities, and I'm not showing here all the data, obviously, many similarities with mammals, like presence of multipotent radial glial cells, presence of intermediate progenitors, production of different types of neurons that are are produced sequentially over time and that take different positions in these layers. And so that pushes us to think that there are conserved principles of cortical development, but it also gives us a more refined view of what is different again, in a mouse neocortical system, because there are some differences, and these are definitely innovations in the mammalian line of evolution. And one difference is this sequence of neurogenesis. In non-mammals, that's true also for reptiles, the early-born neurons are superficial, and then late-born neurons are deeper. But in mammals, there's been some invention, some mechanism that enables the late-born neurons to migrate past the early-born neurons and occupy more superficial layers. And the other innovation is, of course, that in mammals there are more cell types. But given that we see that at the progenitor levels things are very similar and comparable, at this point what we believe is that this greater diversity of neuron types in mammals, the 6 layers, is about the genes and the gene regulatory networks that specify neurons after cell cycle exit, after that progenitor state. Because progenitors, at least with the analysis that we have done, don't display dramatic differences in their molecular identities to explain why you have more and more diverse neurons in a mammal, in a mammalian neocortex, rather than in the salamander. So to sum up this part, point number 3 is that we see that greater neuronal diversity may still evolve in the context of these largely conserved developmental programs, so early development being more constrained, and then as development progresses, these constraints being relieved to enable more diversification of neurons once these progenitors exit their cell cycle. Okay, so this was a lot. In the last few minutes, I want to move a little bit away from cortex to try to illustrate, again, a couple of other principles, but also how some of these ideas may generalize. And so far, I've been telling you about these brain regions. I've been comparing the neocortex to the dorsal pallium and dorsal cortex in reptiles and amphibians. I've not told you much about birds. It's not our work, but there's work in the field, in the community, that is coming to similar conclusions to what we are finding. But there is another very important piece of the brain to consider here, which is another part of a telencephalon that we call ventral pallium, and that in reptiles and birds takes the name of DVR. DVR stands for dorsal ventricular ridge. You see a question mark on the mouse. It's because we still don't really know what it really corresponds to in mammals. But this is a prominent region of the brain in reptiles and birds, and especially in birds has been heavily studied because this is where all the sensory a lot of sensory processing occurs. This is the brain region that is involved in the song system. It has been very heavily studied in the songbird. And a very popular idea that goes back to 1969 with the great work of Harvey Carten is that there are the same circuit motifs in the DVR of birds and the neocortex of mammals. And here with circuits, the definition has been there are input receiving neurons from thalamus that receive sensory information. There are processing neurons that process this sensory information. And then there are output neurons that control some motor programs outside of the telencephalon. And so when we got our first turtle data, we thought we could use this comparison to test whether that idea was supported by cell type data, because you can imagine that this similarity of circuits is a result of homology, like the same neuron types that exist in these different structures. But that doesn't really fit with the fact that one structure is ventral and one structure is dorsal. Or you could imagine that these similar circuit motifs have evolved independently. We call this convergence in these different animals. And so when we did our comparison with turtles, we essentially wanted indeed to test this hypothesis in homology versus convergence, with the idea that if the cells had similar effector genes and the same transcription factors, that would mean that the genetic instructions to make those neurons were the same in the two systems, so that would be evidence for homology. But if we were to find similar effector genes but different transcription factors, that would have been an evidence for the use of different genetic instructions to turn on similar genes, and that's really a classical case for convergence. And so this was a very old analysis limited by the data that were available at the time, but what essentially we found in the turtle is that this DVR of turtle, when you look at all the genes, the transcriptome of this DVR correlates very nicely with the neocortex. And when we look at all the genes, the large majority of those genes are the effector genes because they're way more than transcription factors. But then when you compute these correlations by using transcription factors alone, you see that the correlations are completely gone. So you still have correlation of the cortex, which is the part I've been talking to you about for most of the presentation, to neocortex, because there's some more similarities there. But with the DVR is essentially using transcription factors that have nothing to do with what's being used in the mammalian neocortex. So that's really the classical case for convergence, and this has been repeated with the bird data after us in a way that's more sophisticated and corroborated very extensively. But then we wanted to ask, okay, where is this DVR coming from? And so that we compared, at that point we were prompted to compare the salamander to the reptilian data. And in the interest of time, I will skip to the conclusion of this, which is that, yes, indeed, there are some cells in the salamander, so-called ventral pallium, which have molecular profiles that are very similar to cells in the DVR of reptiles, indicating that these regions may actually trace back to tetrapod ancestors. And so putting this all together, this analysis confirmed the idea that these circuits and circuit motifs evolved convergently in birds and in mammals. But we've also done tracing experiments to look at connectivity in the amphibian, and we saw that there are some projections from the thalamus to this region, which has a molecular profile similar to the DVR of reptiles. So it could be that these circuits were first in the ventral pallium, and then again in the mammalian line of evolution, some of those elements were co-opted and recruited to the dorsal pallium, to the expanding neocortex, for more sophisticated or expanded sensory processing. So that's where we are right now, but of course there's a few pieces still missing. And the bigger, more broader take-home message is that similar neural circuits may evolve by convergence. The last point that I want to make is, do these principles extend to the rest of the brain? And I want to talk here about this assumption that there are certain brain areas that are more conserved than others. There is this theory or hypothesis that we have a reptilian brain inside, and then there is a mammalian brain and a primate brain. It's, of course, evolution doesn't work like that, and no serious neuroscientist would subscribe to that view. But there is nevertheless this idea that is very much ingrained that there are certain brain areas that are more ancient than others. Like the hypothalamus is evolutionary ancient. You see, I'm just taking pieces from publications. And we, in light of what we've seen with the neocortex, we thought there was probably more reasons to challenge that. And so we've collected, when I was still in Frankfurt, a large data set from lizards, spanning almost the entire brain except the hindbrain. And then we've compared these transcriptomes and cell types to mouse data that were, at the time, the best match in terms of sequencing depth and resolution. And what I'm going to show you in a second is a matrix where I show for each lizard cluster, how many cells of that cluster map to mouse cells. So essentially, you can think of it as a similarity matrix. And so the matrix on top here is the whole matrix of all the cell types we compared in mouse and lizard. And this is a filtered down version with the best matches, the highest correlations. So the takeaway from here is that ear cells are sorted by brain regions. So you have a telencephalon here, and then you have more posterior brain regions here. And the takeaway here is that there's not like one single brain region where you have massive conservation of cells and other brain regions where you have less conservation. You find punctuated cell conservation all over the brain. And this is quantified here. And it's actually interesting that even if you look at the distribution, there seem to be even fewer conserved neurons and clusters in diencephalon, for example, rather than in telencephalon. So it's a way that it changes completely our way to think about brain evolution from the concept of this region is more conserved, this region is less conserved, to a more granular view where it's about the cells in each region, and the idea that neuronal diversity can expand and evolve everywhere in the brain. And that's important, I think, because it changes also the kind of questions and hypotheses that we want to ask going forward. Because if it's not about the brain region, but about the cells, what if, for example, there are cells that are so conserved because they are connected to one another across these different regions, and cells that are less conserved because these are the new pieces, the new elements of circuitry that have evolved to confer animals to some new properties. So thinking, going away from that brain region bias view is forcing us to think more about neural circuits and systems. And this is where we want to take our work. And essentially, that's what my lab has been building on, trying to build on, to do. In the last 5 years, I would say half of the lab is not doing what I just presented to you, but is rather trying to develop this system the salamanders have been talking about as a proper neuroscience system where we can start to ask questions about how are these neurons connected and what do these neurons do. And I think we are in a very good position to do that now because of all the development of viral tools, calcium imaging and behavioral assays that we're recently being able to achieve, and some of this is also already published. So I want to thank my lab for being brave and curious explorers of the brain. The work I've been presenting is mostly, it's work of many people, actually. So the single-cell work on salamanders being done by Jamie, Alonso, Eliza, Eli, and Astrid. Astrid has been also the one who really drove the development project I've been telling you about. Many of the things I've been describing are also included the work that I've done as a postdoc at the Max Planck for Brain Research with Gilles Laurent. I want to thank all our collaborators, our funding sources, and again, thank you for this opportunity today.
So I have a question I feel kind of guilty asking because you showed all this incredibly beautiful data about neuronal cell types, but I kept wondering about non-neuronal cell types. So did you look at non-neuronal cell types across, and do you see sort of similar principles, or is it or can you speak to that?
So we looked at them. Obviously, we're actually looking a lot at astrocytes because we have some projects in the lab about regeneration, and astrocytes or astrocyte-like cells are the ones engaged into that. But you have, microglia, you have oligodendrocytes, oligodendrocyte precursor cells, all beautifully, nicely conserved and aligned, all the way to the ancestor of jawed vertebrates. Things are a bit weird with lampreys. But when it comes to astrocytes, we see in many, in reptiles and in amphibians, we see cells that are called ependymoglia cells that essentially express gene modules of mammalian astrocytes and mammalian human stem cells. So one way to look at it is that these cells essentially are functionally astrocytes. They do all the things astrocytes do, but on top of that, they're also able to sustain adult neurogenesis, which is widespread in the brain. And so they rarely but slowly they divide. And then if you do an injury to the brains of these salamanders, they boost divisions like crazy and then reconstitute the lesioned area. Yeah.
Hi. I was reading about some research, fMRI research on the human brain regarding language processing. And what it showed was that there are different regions that light up for nouns and others that light up for verbs. So I'm wondering if there's any kind of equivalent, or I don't know if that's the right word, for such brains. Where would these humans regions evolve from?
Yeah, so I'm not a specialist on this. I think this is rather something to ask to people that are interested in what's new about the human and the primate brain, which is a topic on its own. We know that the ancestral mammal had a 6-layered neocortex, but again, it was much smaller than the neocortex of primates and humans. There's been a great expansion of these neocortices. One idea is the idea that as you expand this cortical tissue, you can essentially, again, repurpose some of this for new functions. So one possibility, one hypothesis is that this language processing area evolved from neocortical areas that were doing other things, and then it started to be involved in new functions. So being the architecture more or less conserved, but the upstream and downstream connections to these cortical areas are different so that the new functions could be implemented.
Really cool talk. So, so you were talking about this idea that different brain regions might have, changed more compared to other brain regions, and you're challenging some of that with. So, if you go even broader and look at like different organs in the body, is it true that brains have changed much more than lungs or other organs among different species?
You find me unprepared, Vikram. Honestly. I guess I'm speaking without being super informed. My sense is that, yes, there's more diversity in the brain rather than in other organs. But if you were to look outside of vertebrates, you will see more diversity also in other tissues. We know that the organs as we know them of invertebrates do not necessarily exist in invertebrates. There's been evolution of the gut and of the heart and of other things. But yeah, it's an interesting question. I'm not prepared to that.
Thank you so much for the talk. So my question is a little bit similar to the previous question, but more particular to like retina, because retina is also a part of the brain tissue, and I feel like I think this salamander has like same number of layers like with mice. So what do you think about what's happening?
I'm not a specialist on this, but there's been a very, very nice study on the evolution of vertebrate retina last year, where retina of many, I think, 17 or 19 vertebrate species have been compared. And the concept there is sort of similar to what I presented in the sense that the main classes of cells seem to be there in all vertebrates, but there are some specific subclasses that seem to be specific of certain lineages of vertebrates. And please correct me if I'm wrong with that. And there seems to be incredible diversity, especially for the retinal ganglion cells. There's lots of different types, and apparently lots of evolutionary divergence there, so which I think is very interesting.
Thank you, Maria. I was struck by the inside-out versus outside-in difference, and it looked like the sort of cell layer was on the inside of the salamander. I'm thinking back to things about the cephalopod where like the gastrulation goes on the opposite direction. Do you know what the significance of the inside-out versus outside-in development does, and what impact it may have? Like, do the outside-in regenerate and the others don't.
Yeah. I don't know because nobody knows, right? These are the hard questions, that I think without a time machine, we will never be able to answer. But I can speculate. And so what I did not have time to present, but what we see is that this dichotomy of early born versus late born neurons aligns also to the kind of projections that these neurons have, connections that they have with the rest of the brain. So we see that the early born neurons of the salamanders are extratelencephalic projection neurons, in the same way the early born neurons of the mouse are extratelencephalic projection neurons. And so the other class is mostly intratelencephalic projecting. So the speculation here is that by switching the order, you enable the expansion of the intratelencephalic projection layer. And indeed, this expansion it's very clearly there when you compare across mammals. If you look at this superficial layer 2-3, and you compare rodents to different primates, you see that it gets bigger, larger and larger, thicker and thicker as you look at other, in the primate lineage. And that's because if the cortex expands overall, you need more neurons to interconnect all these different cortical areas. So you need more of these intratelencephalic neurons, and you need to pack more of them. So the speculation is that the inversion facilitates the expansion of the intratelencephalic, which is necessary for the expansion of the cortex overall. Okay.
So did you look at splice variants and microRNAs? Are they also are they conserved across species, or do you see any difference in things like that? I know you saw transcription factors are different, but I assume microRNAs are more conserved, somewhat relatively. I think.
No, we've not. It's an interesting question. We wondered about that at some point. It's the microRNAs specifically; they're not captured by these single-cell techniques, so we could not look at that just because. Practically, we don't have the data, we don't have the information.
And what about the splice variants? Because there's always reports about, certain variants are doing Alzheimer's or something else. And so I was wondering if there's anything across.
We've not looked at that, and I'm not aware of anyone looking at this question in the context of vertebrates, the way we've been looking at it.
Thank you.
It's been shown that indeed splice variants are differentially expressed in different neuron types. So that's indeed an interesting point. Again, the technology does not enable us to look at this.